Hello, friend. Welcome to Science for Sleep. Whether you have returned for another quiet night or have only just found this channel, this is a gentle place to slow your thoughts, learn something peaceful, and allow the day to fade into the distance. Tonight, we are beginning with a tortoise named Jonathan. He lives on the grounds of Plantation House on St. Helena, a small volcanic island rising from the South Atlantic Ocean. The lawns around the house are green and open, with low shrubs, patches of shade, and familiar paths worn through the grass. Jonathan moves through this landscape at an unhurried pace. His shell is broad and weathered. His legs are thick. His neck stretches forward toward food, and each step places his weight carefully onto the ground. Nothing about the scene appears hurried or dramatic. Yet the animal crossing this lawn may have hatched around the year 1832. That would make Jonathan at least 194 years old in 2026. No written record marks the day he left his egg, so his age had to be reconstructed from the evidence that remains. Jonathan was brought to St. Helena from the Seychelles in 1882. Before we follow that evidence any further, if you enjoy these quiet journeys through science, you are welcome to subscribe. And if it feels natural, you can leave a comment sharing where you are listening from tonight. It helps other curious listeners find their way to this calm corner of the world, while the quiet simply continues here as it is. A photograph taken only a few years after Jonathan arrived shows him standing beside another tortoise near Plantation House. He was already fully grown. Because a Seychelles giant tortoise can take many decades to reach its full adult size, the people who studied the photograph estimated that Jonathan was at least 50 years old when he arrived. That placed his birth around 1832, and it may have been even earlier. Jonathan's age becomes easier to picture when it is placed beside the world that changed around him. When he was probably a hatchling, the practical electric light bulb had not yet been invented. Alexander Graham Bell would not receive his telephone patent until 1876. The first controlled powered airplane flight would not take place until 1903. Penicillin would not be discovered until 1928. Jonathan was already an old tortoise by the time many inventions now treated as ordinary first appeared. Human generations came and went. Ships changed from sail to steam and then to engines powered by oil. Messages that once crossed oceans in weeks began traveling through cables, radio signals, satellites, and the internet. Through all of it, Jonathan continued carrying the same living body. His heart kept moving blood. His lungs kept exchanging gases. His digestive system kept breaking down grasses, leaves, fruit, and vegetables. His immune system kept responding to injuries and infections. Inside his cells, damaged molecules had to be repaired, replaced, or removed again and again. A lifespan of nearly 2 centuries is not one long act of survival. It is an enormous number of ordinary days during which thousands of biological systems continued doing enough of their work correctly. Jonathan is not untouched by age. His eyesight has faded, and his sense of smell is weaker than it once was. But he still responds to familiar sounds, feeds with help from his caretakers, and moves through the same grounds where he was photographed in the 19th century. His survival presents a question much larger than one famous animal. Was Jonathan simply fortunate enough to avoid the accidents and diseases that end most lives? Or does the tortoise body itself change with age in a way that is fundamentally different from the bodies of humans and many other animals? A number such as 194 tells us how long a life has lasted. It does not yet tell us how quickly that body has aged. A long life can come from several advantages working together. Jonathan has lived in a protected setting on St. Helena, with regular food, attentive caretakers, and veterinary help when his health has weakened. He has also avoided severe injury, destructive storms, major infections, and the many accidents that can end an animal's life before old age arrives. Those fortunate conditions help explain how a tortoise survives for so many years. They do not reveal how quickly his organs, tissues, and cells have changed during that time. Scientists therefore separate lifespan from the process of aging. Lifespan is the total length of one individual life. For Jonathan, that measured span has reached at least 194 years. Life expectancy describes something broader. It is the average length of life expected for members of a population under a certain set of conditions. A hatchling living on an island filled with introduced rats may have a very different life expectancy from a hatchling protected inside a breeding center, even when both belong to the same species. Aging researchers also use the word senescence. Senescence means the gradual decline in the way a body functions as age advances. Cells may repair damage less efficiently. Immune responses can become weaker or poorly controlled. Muscles may lose strength, and organs can become less able to recover from stress. Researchers study these changes directly when possible, but they also examine a simpler result that can be measured across large groups of animals. They ask how the chance of dying changes with age. Imagine following thousands of animals from early adulthood onward. At each age, scientists calculate the chance that an individual will die before reaching the following year. For humans, that risk rises sharply during later life. A healthy 30-year-old has a much lower chance of dying within the next year than a healthy 90-year-old. Placed on a graph, the human mortality line remains fairly low through early adulthood and then curves upward more steeply as the decades pass. That rising curve is one clear sign of senescence. Many parts of the body have lost some of their earlier ability to resist disease and recover from damage. A very different pattern appears in certain turtles and tortoises. Their yearly risk of death may change only slightly across much of adulthood. A 60-year-old tortoise can sometimes have a survival chance surprisingly close to that of a 30-year-old adult from the same population. Scientists describe this pattern as slow senescence, when the increase is small. The term negligible senescence is used when the increase is so slight that researchers cannot clearly detect it in the available records. Negligible simply means too small to measure with confidence. A weak increase may still exist. Long-lived animals can also show decline in one part of the body, while their overall mortality risk remains low. Jonathan's cataracts provide a useful example. His eyes have aged enough to cause serious vision loss, while the rest of his body has continued supporting movement, feeding, circulation, and survival. In 2022, researchers examined records from 52 turtle and tortoise species living in zoos and aquariums. These facilities often keep detailed notes about birth dates, transfers, health, and death. That information allows scientists to compare survival at different ages more accurately than they often can in the wild. Roughly 75% of the species in the study showed slow or negligible senescence. This pattern appeared across many kinds of shelled reptiles, rather than only among a few famous giant tortoises. Such animals remain fully vulnerable to real dangers. A severe infection can become fatal. Lack of water or food can overwhelm the body. Falls, floods, freezing conditions, habitat destruction, and organ disease can still end a life. Their unusual pattern lies in the effect of age itself. For many species, another passing decade adds far less mortality risk than it would for a mammal. That changes the mystery surrounding Jonathan. His record involves more than extraordinary luck or careful protection. He belongs to an ancient group of animals in which adulthood can remain surprisingly stable over very long periods. The origin of that stability reaches far beyond one old tortoise on St. Helena. It begins within the body plan shared by tortoises, freshwater turtles, and sea turtles, assembled over hundreds of millions of years. The group that contains tortoises is called Testudines. This reptile order includes land-dwelling tortoises, freshwater turtles, and sea turtles. They all share the same basic feature: a body enclosed by a shell that is joined directly to the skeleton. The word tortoise usually refers to members of this group that spend nearly all of their lives on land. Their bodies have been shaped by the demands of walking, carrying weight, finding water, and surviving without the support of a lake or ocean. A giant tortoise may weigh more than 200 kilograms. That mass rests on 4 thick limbs that rise beneath the body like short pillars. Each foot is broad and sturdy, with strong claws and compact toes suited to pushing against soil. The animal's slow walk comes from a design made for support and endurance. Freshwater turtles often have flatter feet with webbing between the toes. Webbing increases the surface area that presses against water, making each stroke more effective. Sea turtles have taken that aquatic design much farther. Their front limbs have become long flippers that move through the ocean like wings. The shell is lower and more streamlined, allowing water to pass over it with less resistance. A sea turtle's head and flippers remain partly exposed because the body shape required for long-distance swimming leaves less room for complete withdrawal. A land tortoise follows a different physical path. Its shell can rise into a high dome. The limbs bend and pull inward when danger approaches. Many species can bring the head beneath the front edge of the shell, placing soft tissues behind bone and tough scales. This protective shape adds weight and creates drag in water. Yet those costs matter less for an animal living on dry ground. Tortoises still share deep ancestry with every other turtle. Their land adaptations were added gradually to a body plan that had already existed for millions of years. Evolution works by changing structures inherited from earlier generations. A limb that once helped an ancestor move through shallow water can become thicker and better at supporting weight. A flatter shell can become taller when swimming speed no longer controls survival. Small changes in bone growth, muscle attachment, and body shape accumulate across very long periods. The giant tortoises of the Galápagos Islands and Aldabra Atoll show how much can happen after related populations follow separate paths. The Galápagos lie in the Pacific Ocean, about 1,000 kilometers west of mainland Ecuador. Aldabra sits in the Indian Ocean, more than 15,000 kilometers away. The tortoises living in these two places look similar because both became enormous, slow-moving island herbivores. Genetic evidence shows that their lineages last shared a common ancestor roughly 40 million years ago. That ancestor lived long before modern humans appeared. The line leading toward giant tortoises had already separated from the lineage leading toward humans more than 300 million years ago. During that immense stretch of time, the two giant tortoise groups evolved on different branches. Their large size and unusual longevity, therefore, did not emerge from one recent island population that simply spread across the world. Each lineage developed those qualities within the much older turtle design it had inherited. The shell existed before the first Galapagos giant tortoise. It also existed before the first Aldabra giant. Protection was already built into the basic structure of the group, creating a strong starting point for animals that would later grow slowly and survive through many breeding seasons. What appears from the outside as a hard covering reaches much deeper into the body. The ribs have changed shape. Parts of the spine join the upper shell. Muscles pass through an arrangement found in no other living vertebrate. Even the shoulders sit in an unexpected position, enclosed within the framework formed by the shell. That hidden arrangement begins with a common mistake. A tortoise does not carry its shell like a house that could be left behind. The shell is part of the skeleton. It is connected to the spine, joined with the ribs, supplied with blood, and covered by living tissue. As a tortoise grows, the shell grows with it. The upper part is called the carapace. The lower plate beneath the body is the plastron. Together, they form a strong frame around the chest and abdomen. The carapace developed mainly from ribs that became wider and flatter over millions of years. Parts of the backbone also became locked into this upper shield. The plastron has a different origin. Some of its bones formed from structures related to gastralia, a set of small belly bones found in several ancient reptiles. These bones gradually enlarged and joined together beneath the body. The result is a skeleton, unlike the frame inside a lizard, bird, or mammal. The shoulders show the difference especially well. In most animals with 4 limbs, the shoulder girdle sits outside the rib cage. You can picture the upper arm attaching near the outer wall of the chest. Turtles develop in another way. As the embryo grows, the ribs spread outward, and the shell forms around the shoulder region. The shoulder girdle ends up inside the border created by the ribs. That unusual arrangement confused early anatomists because it seemed to reverse the normal plan of a vertebrate body. Fossils reveal that the change happened in stages. One of the earliest important animals is called Untosaurus africanus. It lived in what is now South Africa about 260 million years ago. Untosaurus had broad, flattened ribs, but it did not yet have a complete turtle shell. Its back would have looked wider and more rigid than that of an ordinary reptile. Later, relatives developed larger bones across the belly. By around 220 million years ago, a turtle relative named Odontochelys had a well-developed plastron beneath its body. Its upper shell was still incomplete. This means the lower armor appeared before the fully enclosed carapace familiar today. Other fossils show further steps as the ribs widened. The back became more solid, and the shell edges closed around the body. The complete design took tens of millions of years to assemble. Rib broadening may have begun roughly 50 million years before the first fully formed turtle shell appeared. Those early changes brought serious costs. Wide ribs make the trunk stiff. A stiff body cannot bend from side to side as freely while walking, so the stride becomes shorter. Most reptiles also use movement of the ribs to expand the chest and draw air into the lungs. Once turtle ribs became locked into the shell, they could no longer move in the ordinary way. Modern turtles breathe with special sheets of muscle inside the body. These muscles shift the organs and change the space around the lungs, drawing air in and pushing it out. The shell therefore changed movement and breathing long before it became the polished shield seen on a living tortoise. Researchers have suggested that the earliest widened ribs may have helped with digging. A stiff trunk can give strong front limbs a solid base. Unitosaurus had features linked with burrowing, including powerful forelimbs and claws. The first stages of shell evolution may have supported an animal pushing through soil. As the ribs and belly bones continued expanding, the same structure became more useful as armor. A complete shell protects the spine and shields many internal organs. A tortoise can draw vulnerable tissue behind the hard edges, leaving a predator to face bone covered with tough keratin. Keratin is the same durable material found in human fingernails and animal claws. By the time the shell was complete, the turtle body had accepted slower movement and a more complicated way of breathing in exchange for remarkable protection. An adult equipped with that defense had a better chance of surviving attacks that would quickly kill a softer-bodied animal. Across many generations, that higher survival rate began to change how slowly a turtle could afford to grow and how much energy it could invest in maintaining the same body for years to come. Imagine a small animal living in a place where predators find it easily. Even with excellent DNA repair and strong organs, it may be eaten before its first birthday. Energy spent preserving that body for several decades would bring little benefit if most individuals never survive long enough to use it. Now picture an adult tortoise protected by a thick shell. A predator may bite, claw, or push against the hard surface and eventually move away. The tortoise survives, recovers, and reaches another breeding season. That difference matters across many generations. Natural selection works through traits that help organisms leave surviving offspring. When adults regularly live for many years, features that preserve their bodies can improve reproductive success long after early adulthood. Biologists use the term external mortality for deaths caused mainly by outside forces. Predators are one source. Falling rocks, severe storms, and accidents can also end a life before internal aging becomes important. High external mortality reduces the chance that careful long-term maintenance will ever be rewarded. A body that may disappear quickly often benefits from growing fast and reproducing early. A well-protected animal can follow a slower schedule. Scientists call this schedule a life history strategy. It describes when an animal grows, when it begins breeding, how often it reproduces, and how long it is likely to survive. Every organism has a limited supply of energy. Food provides that energy, and the body must divide it among many tasks. Growth requires new bone, muscle, and tissue. Reproduction demands eggs, sperm, courtship, and movement toward nesting areas. Immune defense consumes resources when the body responds to infection. Cellular repair replaces damaged parts and corrects mistakes inside DNA. Stored fat and water help the animal endure periods when food becomes scarce. No animal can spend unlimited energy on every task at once. A short-lived species may place more of its available energy into rapid development and early reproduction. A species with high adult survival can spread those costs over a longer life. Slow growth becomes possible because the animal has a good chance of reaching maturity. Delayed breeding becomes less risky because many adults remain alive long enough to reproduce repeatedly. Maintenance also gains value when repaired tissues may support dozens of future breeding seasons. This connection has been tested across many kinds of reptiles and amphibians. In 2022, researchers compared 107 wild populations, representing 77 species. The animals included turtles, snakes, lizards, frogs, salamanders, and crocodilians. Some possessed shells or thick armor. Others carried spines or produced chemical defenses that made them difficult or unpleasant to attack. The researchers compared these protective traits with measured rates of aging and adult longevity. Species with stronger protection often aged more slowly or lived longer. Turtles were among the groups showing especially low rates of adult aging. Their shells had reduced danger from predators for millions of years, giving natural selection more opportunity to favor traits that preserve the same body. The pattern does not mean that armor alone creates a long life. Many protected animals still die young. Disease, drought, poor nutrition, and habitat loss can overcome even a strong shell. Body size, climate, reproduction, and evolutionary history also influence survival. The study showed an association across many species rather than a simple rule that applies in every case. The shell also has no direct control over the enzymes that repair DNA inside a cell. Its influence works through survival. Adults that repeatedly escape attack gain more chances to reproduce. Lineages that maintain healthy tissues for longer periods can then leave more descendants. Across thousands of generations, protection and bodily maintenance can begin reinforcing each other. This helps explain why the tortoise body follows such a patient schedule. The shell made a long future more likely, allowing evolution to invest in animals that could use that future. Maintaining a body for decades still carries a cost. Every repair, heartbeat, and movement requires energy, and a tortoise gathers and spends that energy under the changing warmth of the sun. A tortoise begins each day with a body temperature closely tied to the world around it. Biologists call this condition ectothermy. An ectotherm depends strongly on outside heat to warm its tissues. After a cool night, a tortoise may remain still until sunlight reaches the ground. It can turn the broad surface of its shell toward the morning sun, allowing heat to pass into the skin, blood, and deeper tissues. As the day grows warmer, the animal may move beneath a shrub or settle against cooler soil. Some giant tortoises rest in shallow pools or muddy wallows when the air becomes hot. Each choice changes the temperature of the body without requiring a large internal furnace. Mammals and birds follow a different energy pattern. Their cells release heat continuously, keeping the body within a narrow temperature range through cold nights and changing weather. A human body usually remains close to 37 degrees Celsius. Maintaining that temperature requires a steady supply of food and oxygen, even during sleep. Tortoises spend far less energy producing heat inside their bodies. That saving comes with a close dependence on sunlight, shade, wind, and the temperature of the ground. A cold tortoise moves slowly because chemical reactions inside muscle cells are proceeding at a reduced rate. Signals travel through the nerves less quickly. Blood circulates more slowly, and the muscles produce less force. Digestion also changes. After eating grasses, leaves, or cactus pads, a tortoise needs sufficient warmth for the digestive system to process the meal efficiently. Food may remain inside the gut much longer during cool conditions. Immune activity is also influenced by temperature. White blood cells identify damaged tissue and respond to infection through chemical reactions that work best within certain temperature ranges. A tortoise may sometimes seek warmer places while ill, using behavior to raise its temperature in a way that resembles a fever. The animal therefore regulates many body functions by moving through the landscape. A few meters between open sunlight and deep shade can produce a meaningful change. Posture also matters. Stretching the legs and neck exposes more skin to warm air. Pulling inward reduces the amount of soft tissue exposed. The shell itself absorbs heat slowly and releases it gradually, especially in a large adult with considerable body mass. This daily pattern can make tortoise biology appear uniformly slow. Metabolism actually changes throughout the day. Metabolism is the full set of chemical reactions that keeps an organism alive. It includes extracting energy from food, building new tissue, and repairing damaged cells. A warm, active tortoise uses energy at a higher rate than the same animal resting in cool shade. Body size changes the calculation as well. A 200-kilogram giant tortoise handles heat differently from a small desert tortoise weighing only a few kilograms. Species living in tropical lowlands also face conditions unlike those experienced by tortoises that spend winter inside underground burrows. This variation matters when thinking about aging. Researchers comparing reptiles and amphibians in 2022 found an enormous range of aging rates. Several turtle species changed very little across adulthood. Other ectotherms showed much faster decline. Some frogs and lizards lived only a few years, despite relying on environmental heat in the same broad way. Ectothermy can reduce the constant cost of maintaining a warm body. It can also lower activity during periods when food and water are scarce. Those advantages contribute to a flexible energy budget. Longevity still depends on the protection of the shell, adult size, patterns of growth, habitat conditions, and the ability of cells to maintain themselves. The sun helps set the pace of a tortoise's body from hour to hour. Survival across decades depends on how that changing supply of energy is divided among movement, digestion, growth, and repair. On a cool morning in the Galápagos, a giant tortoise may wait several hours before beginning to feed. The air near the ground is still cool, and the muscles inside its heavy limbs cannot yet work at full strength. As sunlight warms the shell and surrounding soil, chemical reactions inside the body begin moving faster. By midday during a cooler season, the animal may be walking through grass or feeding on low plants. Hot weather creates a different schedule. Movement often begins earlier, while the ground still holds some of the night's coolness. Feeding may slow during the hottest part of the afternoon. A tortoise can rest beneath shrubs, settle into damp soil, or lower its body into a muddy pool. Activity may return later, once the sun has dropped and the air begins to cool. These changes allow the animal to use warmth when it is helpful, and avoid spending water during dangerous heat. Galapagos giant tortoises commonly rest for about 16 hours during a day. Some of that time is spent asleep. Long periods are also passed quietly beneath vegetation, or with the limbs folded beneath the shell. During drought, stillness can last even longer. Walking across dry ground costs energy. It also increases water loss through breathing and exposure to heat. When fresh plants are scarce, traveling farther may produce little reward. Remaining in one sheltered place can be the better choice until rain returns or new growth appears. The body continues working throughout these quiet hours. The heart keeps blood moving through vessels that reach the organs, muscles, and living tissue beneath the shell. Nerve cells maintain small electrical differences between their inner and outer surfaces. These differences depend on charged particles called ions, including sodium and potassium. Without that careful balance, nerves could not send signals and muscles could not contract. Bone also remains active. Old material is slowly removed while new material is added. The shell contains living bone beneath its outer keratin covering, so it must be maintained along with the rest of the skeleton. Immune cells continue watching for damaged tissue and signs of infection. Food may still be moving through the digestive tract many hours after it was swallowed. A resting tortoise therefore uses less energy than an active one, while still supporting a large number of essential processes. Feeding follows the same flexible pattern. Galapagos tortoises eat grasses, leaves, and native fruit when these foods are available. In dry areas, cactus pads can provide both moisture and energy. The animals may also consume fallen flowers or low shrubs. They have no teeth. A hard, curved beak cuts plant material into pieces that can be swallowed. Digestion happens slowly, helped by microbes living inside the gut. These tiny organisms break down tough plant fibers that the tortoise could not fully process on its own. Rain can quickly change the value of a landscape. Shallow pools appear in low places. Dry plants produce softer new growth. A tortoise reaching fresh water may drink a large amount at once. Some water can be held in the bladder and used later as conditions become dry again. This storage does not give every tortoise a guaranteed number of months without drinking. Survival depends on body size, temperature, recent feeding, health, and the dryness of the habitat. A large adult with full reserves can usually endure scarcity longer than a small juvenile. Energy conservation also reduces the need to search constantly for food. Fewer unnecessary movements mean fewer falls, less exposure to heat, and lower use of stored water. Reduced activity may limit some forms of wear inside the body, although longevity cannot be explained by stillness alone. Repairing tissue and supporting immunity still require energy. The advantage comes from matching effort to opportunity. Warmth is used when movement and digestion will pay off. Rest becomes useful when the landscape offers little food or water. Across one day, this strategy may look like simple slowness. Across many dry seasons, it becomes a reliable way to remain alive until conditions improve. The full scale of that patience appears in the time needed for a hatchling to become a giant adult, building its shell and body over several decades. A giant tortoise begins life small enough to fit inside 2 human hands. Its shell is already present, yet the young body has little of the strength seen in a full-grown adult. The bones are thinner, the muscles are smaller, and the amount of stored water and energy is limited. A hatchling crossing open ground can be threatened by animals that would barely concern an adult tortoise. Its future protection must be built slowly from food, minerals, sunlight, and time. The shell expands along with the rest of the skeleton. Living bone beneath the outer surface is constantly being remodeled. Some old bone is broken down, while fresh material is added in its place. Keratin forms the visible scutes covering the shell. Keratin is the tough material also found in claws and human fingernails. As the tortoise grows, new keratin develops around and beneath older areas, allowing the shell to widen and thicken. The internal organs must enlarge within that hard framework. Leg muscles become strong enough to lift increasing weight. The lungs, heart, digestive tract, and blood vessels develop to support a body that may eventually weigh hundreds of kilograms. This construction continues long after many other animals have reached adulthood. Galapagos giant tortoises usually become sexually mature at around 20 to 25 years of age. A house mouse can produce young only a few months after birth. Many wild birds begin breeding within their first or second year. A giant tortoise may spend more than 2 decades growing before producing its first offspring. That delay becomes possible when adults have a strong chance of surviving far beyond maturity. Body size brings several practical advantages. A large tortoise can store more fat and water than a hatchling. Its temperature changes more slowly because a heavy body takes longer to warm and cool. This added stability helps the animal move through changes between hot days and cooler nights. Few natural predators can attack a giant adult successfully. Height also improves access to food. A long neck can reach leaves or cactus pads above the level available to smaller animals. Some male Galapagos tortoises exceed 200 kilograms, although size varies among islands, species, and individuals. Males are usually larger than females within the same population. Their shell shapes also vary. Many tortoises from wetter highlands have a rounded form known as a domed shell. The front edge curves downward around the neck, giving the body a high, enclosed shape. These environments often provide grasses and low plants close to the ground. Tortoises from dry lowlands may have a saddleback shell. The front of the carapace rises in an arch, creating more space for the neck to stretch upward. Saddleback tortoises also tend to have longer limbs and necks. These features can help them reach taller shrubs and cactus in landscapes where low vegetation is less dependable. domed and saddleback forms are broad ecological patterns. Some individuals have shapes that fall between the two, and shell form does not reveal every detail of an animal's habitat or behavior. Shape also affects the way a tortoise handles physical danger. A heavy animal can sometimes overturn while climbing rough ground, competing with another tortoise, or crossing a steep slope. Once upside down, it cannot feed or travel toward shade. The exposed soft tissues may lose water rapidly under intense sun. A tortoise that remains overturned for too long can die from overheating, dehydration, or pressure on its organs. Researchers have tested whether shell shape changes the ability to roll back onto the feet. One study created three-dimensional reconstructions from the shells of 89 Galápagos giant tortoises. The models allowed scientists to estimate how far each body would need to rotate and how much energy self-righting would require. A simple prediction suggested that the raised saddleback shape might make recovery easier. The calculations produced a more complicated result. Saddleback shells required more estimated energy to right than the domed shells in the study. Longer limbs and necks may still help a living saddleback push against the ground, but shell shape alone did not provide an easy mechanical advantage. Even a heavily protected body must solve risks created by its own weight. Over several decades, growth transforms a vulnerable hatchling into a massive adult, able to resist predators, store scarce resources, and survive long dry periods. Bodies of this size developed most successfully in isolated places where large land predators were absent and empty ecological roles remained available. Those conditions appeared most clearly on remote islands. The Galápagos archipelago lies in the eastern Pacific Ocean, about 1,000 kilometers west of mainland Ecuador. It is a volcanic chain built over millions of years as the ocean floor moved across a source of rising magma. Some islands are low and dry. Others contain tall volcanoes whose slopes catch moisture from passing clouds. Short distances can separate dusty cactus country from cool highland grass. These differences created several kinds of tortoise habitat within the same archipelago. Populations became divided by stretches of ocean, steep lava fields, and the boundaries between neighboring volcanoes. Over time, isolation allowed separate lineages to develop different body sizes, shell shapes, and feeding habits. A tortoise living in the wetter highlands of Santa Cruz faced conditions unlike those on the dry lowlands of Hispaniola. The Galapagos giants became one of the most familiar examples of island evolution, yet they represent only one surviving kingdom of enormous tortoises. More than 15,000 kilometers away, another population lives in the western Indian Ocean. Aldabra Atoll is part of the Seychelles, north of Madagascar and far from any large continent. Its foundation is coral limestone rather than volcanic rock. 4 main islands form a broken ring around a broad, shallow lagoon. An outer reef surrounds much of the atoll, while channels connect the lagoon with the open sea. The protected World Heritage property covers about 35,000 hectares. Aldabra received that status in 1982, helping preserve one of the least altered large atolls on Earth. Its giant tortoises belong to a lineage deeply separated from the giants of the Galapagos. Their shared ancestor lived roughly 40 million years ago. The two modern populations, therefore, did not arise from a recent journey between the Pacific and Indian Oceans. Each lineage spent an immense span of time evolving within its own island region. Large size can become possible on islands when ordinary continental pressures are reduced. Many continents contain large cats, wild dogs, or other mammals able to attack slow-moving animals. They also support grazing mammals that compete for grasses and leaves. Remote islands may lack those predators and competitors. An arriving turtle lineage can then enter an ecological role that would normally be filled by a mammal. Generation after generation, larger individuals may gain advantages. A bigger body can store more water, reach more vegetation, and resist attack once adulthood is reached. Long isolation gives those changes time to accumulate. On Aldabra, the result can be seen across the entire landscape. UNESCO estimates that approximately 152,000 giant tortoises live there. It is the largest surviving population of giant tortoises anywhere in the world. In some areas, several animals can be seen grazing across the same open ground. Their trails pass through scrub and grassland. Low branches are broken or stripped as heavy bodies move beneath them. Dung carries seeds away from the plants that produce the fruit. Repeated feeding keeps some areas open and changes which species can grow there. This creates an unusual food web. Aldabra has no native large land predator capable of regularly killing healthy adult tortoises. The giant tortoise therefore stands near the top of the terrestrial system as its dominant plant eater. An herbivore occupies the highest major position on land, shaping vegetation through steady feeding and movement. Plants have developed for generations under that pressure. Some grow low enough to be cropped repeatedly. Others survive through tough leaves, rapid regrowth, or branches that rise beyond easy reach. The animals and the landscape have altered one another over long periods. Galápagos and Aldabra show two separate evolutionary paths, leading toward the same broad result. Isolation reduced the danger faced by large adults. Open ecological space allowed turtle lineages to become the main grazers of their islands. Their great size brought protection and storage, while long lives allowed each animal to influence the same landscape for decades. Safety from large predators still left them exposed to dry seasons and uneven food. Giant tortoises answered those pressures through movement, crossing slopes and lowlands as conditions changed across the year. Across Santa Cruz Island, many giant tortoises follow routes that have been used for generations. A visitor watching one animal for an hour might see only a few meters of movement. Over several weeks, that same tortoise can travel between dry lowlands and cooler highland slopes. These journeys connect feeding areas, water sources, resting places, and nesting ground. Santa Cruz rises from warm coastal zones into wetter country at higher elevation. Rainfall increases as moist air moves up the slopes. The highlands support thick grass and other green plants during parts of the year, while the lower regions become hotter and drier. Adult tortoises respond to those changes by moving toward the places offering the best food. During wetter periods, fresh plants may become available in the lowlands. As conditions dry, some animals climb toward the highlands, where moisture lasts longer and vegetation remains more productive. The trip unfolds at tortoise speed. A few hundred meters may take hours. Progress can pause during strong heat. while the animal rests beneath shrubs or waits in a muddy hollow. Movement resumes when temperature and moisture become more favorable. The full route only becomes clear when many small movements are joined together. Researchers have used GPS devices to record those journeys in detail. GPS stands for Global Positioning System. A small tracking unit receives signals from satellites and records the animal's location at regular intervals. Over a 12-year study, scientists followed 25 giant tortoises and reconstructed 140 migration routes. The information revealed repeated paths between elevations rather than random wandering. Some individuals returned to similar areas during the same part of each year. Others adjusted their timing according to rainfall, plant growth, and local conditions. The researchers compared the GPS records with detailed maps of vegetation. That comparison exposed a barrier created by an introduced tree called Cedrela odorata. It is commonly known as Spanish cedar. The species was brought into the Galápagos by people and later spread through parts of the Santa Cruz highlands. Dense stands form areas that giant tortoises often avoid. The trees change the structure of the forest floor and reduce the open routes used by these large animals. Instead of moving evenly through the landscape, tracked tortoises became concentrated through narrow gaps between stands of cedar. A route shaped across many generations could therefore be squeezed into a small corridor by vegetation that arrived only recently. This matters because a protected feeding area has limited value when the path leading to it becomes blocked. Tortoises need connected landscapes. A patch of healthy highland grass cannot fully support an animal that cannot reach it during the dry season. Poor access to seasonal food can reduce body condition, meaning the amount of stored energy and water available inside the body. For an adult female, lower reserves can affect the energy available for producing eggs and completing a long trip to nesting ground. The effects also spread beyond the tortoise itself. Seeds pass through the digestive system and are deposited in dung, sometimes far from the parent plant. Nutrients move with the animal from one elevation to another. Repeated walking presses paths through dense vegetation and creates openings used by smaller species. When migration narrows, those ecological movements narrow with it. Related field programs have followed adult females, hatchlings, and nests across different elevations on Santa Cruz. Researchers record soil temperature, rainfall, vegetation, and the condition of nesting areas. These measurements show that movement and reproduction are tied closely to place. A female feeding in the cool highlands may later travel toward warmer, drier soil where her eggs can develop underground. Her route joins two very different parts of the island. One provides the energy needed to form a clutch. The other provides the heat and soil conditions needed for a new tortoise to begin developing. The slow migration therefore carries more than one adult across a landscape. It carries stored energy toward the place where another life may begin. A female leaving the highlands may spend days or weeks moving toward warmer ground. The route often leads downhill, away from wetter feeding areas and toward dry soil that can hold a nest. In the Galápagos, most breeding activity occurs during the warmer part of the year, although the timing changes from island to island. Nesting often takes place during the cooler, drier months that follow. By then, a female may be carrying eggs formed from energy gathered much earlier. Her journey can cross grassland, rocky slopes, and open ground heated by the sun. Once she reaches a suitable place, she begins testing the soil. The ground must be soft enough to dig, yet firm enough to keep the chamber from collapsing. Moisture also matters. Soil that is too wet can limit oxygen around the eggs, while very dry soil may remove water from the developing embryos. The female uses her hind feet to open the nest. Each foot works behind her body, reaching into a space she cannot see directly. She scrapes loose soil away, shifts her weight, and deepens the chamber a little at a time. This work can continue for hours. When the nest is ready, she lowers the eggs into the hole one by one. They are nearly round and covered by a firm, pale shell. After the final egg is laid, the chamber is closed with soil. The female presses the surface down with her hind feet, smoothing the ground until the nest becomes difficult to notice. Her role then ends. No adult remains nearby to guard the eggs or feed the young after they hatch. The number of eggs varies greatly among Galapagos tortoises. Some saddleback females may lay only 2 to 7 eggs in one nest. Large domed females can sometimes lay more than 20. A single female may make between 1 and 4 nests during a breeding season. This variation reflects differences in body size, habitat, food supply, and the evolutionary history of each population. The eggs remain underground for roughly 110 to 175 days. Development proceeds faster in warmer conditions and more slowly when the nest stays cool. Temperature also helps determine whether the embryos become male or female. Galapagos tortoises do not rely on the same X and Y chromosome system used by humans. Instead, heat affects chemical pathways during a sensitive period of development. Cooler nests usually produce a greater share of males. Warmer nests tend to produce more females. The exact dividing temperature differs among species and depends on the conditions inside the nest, so there is no single number that applies to every giant tortoise. This feature gives conservation teams a useful tool. Inside breeding centers, incubator temperatures can be adjusted to influence the sex ratio of hatchlings. A population with too few females may recover more quickly when more young females are produced. Each one can eventually lay eggs across many future seasons. That future takes a long time to arrive. A Galapagos giant tortoise usually reaches sexual maturity at around 20 to 25 years of age. During those first 2 decades, the animal must survive drought, limited food, disease, and introduced predators. Only then can it begin contributing offspring to the population. The strategy works because adults often remain alive for many years after maturity. A female that survives into her 70s or 80s may have repeated chances to nest even when several seasons fail. Poor rainfall may reduce food and body condition. Flooding can destroy some nests. Predators may take many hatchlings before they have traveled far. Long adult life spreads reproduction across time, allowing later seasons to replace the losses of earlier ones. This makes every mature tortoise biologically valuable. Its body carries decades of growth and survival before the first successful clutch is even possible. Beneath the soil, one of those eggs may contain an animal capable of living for more than a century. Its first years, however, will be the most dangerous part of that entire life. Inside the buried nest, Each hatchling develops within a shell only a few centimeters wide. When development is complete, a small temporary structure on the beak helps break through the egg. The young tortoise may remain underground for a time after hatching. Its body can absorb the last nutrients from the yolk, while the soil above becomes soft enough to cross. Eventually, it begins pushing upward through the nest chamber. No parent waits at the surface. The hatchling enters the open landscape alone, often weighing less than a small household pet. Its new shell follows the same basic plan as the shell of a giant adult. The ribs are joined to the carapace and the lower plastron protects the belly. Yet the young shell remains thin enough for some predators to bite or break. Its legs are short, the neck is small, and the body contains only limited reserves of water and energy. An adult weighing more than 200 kilograms can endure conditions that would quickly overwhelm a hatchling. Danger begins before the egg opens. Pigs can locate nests and dig through the soil. Black rats may eat eggs or attack newly hatched tortoises. Dogs can injure juveniles, while some birds take young animals small enough to lift or peck. Invasive ants may enter nests and attack hatchlings before they fully emerge. Weather adds another layer of risk. A long dry period can leave little soft vegetation near the nest. Small bodies lose water more quickly than large ones because they have more exposed surface compared with their total volume. Limited fat stores provide little protection when food remains scarce for weeks. Heavy rain can also cause trouble by flooding nests or packing the soil above them. These early dangers create a pattern called age-structured survival. The chance of living through the next year changes sharply with age and size. Mortality is concentrated among eggs and small juveniles. Once a tortoise grows large enough, the shell thickens, water reserves increase, and many predators become less dangerous. Pinzon Island offers one of the clearest examples. Sailors introduced black rats to the island during the 19th century. Adult Pinzon tortoises continued living for decades, protected by their size and shells. Almost every new hatchling disappeared. For much of the 20th century, researchers found old adults, but very few young animals. The population still existed, yet it had nearly stopped replacing itself. Biologists describe the addition of surviving young animals as recruitment. Without recruitment, an apparently stable group of adults slowly moves toward extinction. In 1965, conservation workers began transferring Pinzon eggs to protected facilities on Santa Cruz Island. This marked the beginning of the major Galápagos captive breeding and rearing program. Eggs were incubated safely, and the hatchlings were raised away from introduced predators. The aim was temporary protection through the most dangerous stage of life. This method is often called head starting. Young tortoises receive a safer beginning before returning to the wild. By about 5 years of age, many have developed thicker shells and stronger limbs. Their larger bodies hold more water and stored energy. Some populations require a longer stay, depending on local predators and environmental conditions. The first 20 captive reared Pinzon tortoises were returned to their island in 1970. More releases followed as new groups reached a safer size. The deeper problem remained until conservation teams removed the rats. An island-wide eradication effort succeeded in 2012. 2 years later, researchers documented wild-born hatchlings on Pinzon. Young tortoises were surviving naturally for the first time in more than a century. The return of those hatchlings showed how much of the population's failure had been concentrated into one narrow part of life. A century-old adult could continue feeding and breeding, while nearly every offspring vanished. Once the introduced predator disappeared, the same nests began producing survivors again. Passing through roughly the first 5 years changes the balance. The growing tortoise becomes harder to kill, better able to cross dry periods, and more capable of recovering from ordinary stress. Adult survival can then remain remarkably high for decades. The remaining puzzle lies within those long adult years, whether each added decade steadily increases the danger of death, or whether the risk stays almost level across much of a tortoise's life. The steepest danger in a tortoise's life often appears near the beginning. Eggs disappear. Hatchlings fail to find food. Small juveniles remain exposed to predators that cannot threaten a full-grown adult. Once those early years are separated from the long adult period, a very different pattern becomes visible. Scientists study that pattern by calculating mortality risk at each age. Mortality risk is the chance that an animal will die during a certain span of time, often the following year. For a young adult human, that yearly risk is low. It rises gradually through middle age, then increases much more sharply during later decades. A graph of those numbers forms an upward curve. The bend becomes steeper as the body loses some of its ability to recover from infection, injury, and damage inside its tissues. Tortoises can produce a much flatter line. An adult may grow older for several decades, while its measured chance of dying changes only slightly. High mortality among hatchlings does not conflict with this result. The two stages are analyzed separately because a newly emerged tortoise faces dangers that have little to do with aging. A rat taking a hatchling tells us almost nothing about how the organs of a 70-year-old adult are functioning. In 2022, researchers examined survival records from 52 species of turtles and tortoises. The animals lived in zoos and aquariums around the world. These collections included familiar pond turtles, smaller land tortoises, and some much larger species. Researchers used records of births, deaths, transfers, sex, and population size to estimate how mortality changed across adulthood. Managed populations are especially useful for this kind of work. A tortoise in the wild may disappear into dense vegetation, cross into an area that cannot be searched, or die without being found. Its exact age may also be unknown. Zoos and aquariums often keep records for decades. An animal may have a known hatch date, a documented medical history, and a clear date of death. These details allow scientists to compare individuals of different ages with greater confidence. The results were striking. Roughly 75% of the 52 species showed slow or negligible senescence. Senescence is the gradual rise in weakness, disease, and mortality risk that usually comes with age. Negligible senescence means that the increase was so small that the researchers could not clearly detect it in the available data. For many of these species, a much older adult did not appear dramatically more likely to die during the following year than a younger adult. Body size also mattered. Larger species tended to have greater adult life expectancy. A heavy tortoise usually has more stored energy, greater water reserves, and fewer predators capable of attacking it. Those advantages can support long survival even before cellular aging is considered. Some species produced mortality estimates that appeared to decline with age. That result requires caution. The oldest animals in a population may be unusually strong survivors. Small sample sizes can also create unstable estimates. Veterinary care, diet, enclosure design, and differences between institutions may influence which animals live longest. A downward estimate, therefore, does not prove that a tortoise becomes biologically younger as the years pass. The broad pattern remains important. Many turtle and tortoise species lack the steep late-life rise in mortality seen in humans and numerous other mammals. Their bodies still change. Eyes can weaken. Joints can become less flexible, and disease can still develop. A nearly flat mortality curve describes survival across a population. It does not claim that every organ remains in its original condition. Jonathan now fits within a much larger biological pattern. His 194-year life remains exceptional, yet the underlying ability to age slowly appears across many branches of the turtle family. The records also suggested that this aging pattern may respond to surroundings. For some species, safer conditions seem to change the slope of the curve itself. Life inside a zoo or aquarium removes many dangers that shape survival in the wild. Predators cannot reach the animals. Food arrives regularly. Clean water remains available. And injuries can receive treatment before infection spreads. Shelters may protect tortoises from severe cold, intense heat, or long periods without rain. These conditions clearly improve the chance of survival. An animal that avoids starvation and untreated wounds will usually live longer than one facing those dangers every season. Longer life alone does not prove that aging has slowed. Imagine two populations whose bodies weaken at exactly the same rate. One population lives in a protected setting, while the other faces predators and food shortages. More protected animals will reach old age because fewer die early from outside causes. Within both groups, however, the yearly risk of death could still rise at the same rate as adulthood advances. The 2022 study suggested that some turtles and tortoises may show a deeper response. Under favorable conditions, the increase in mortality associated with age appeared to become weaker. Their estimated senescence rate changed along with the environment. Some species seemed able to maintain a flatter survival curve when danger and scarcity were reduced. Biologists use the term phenotypic plasticity for this kind of flexibility. A phenotype is an observable feature of an organism, including body size, behavior, or the way a physiological system functions. Plasticity means that the same inherited biology can produce different results under different surroundings. A tortoise raised with steady nutrition may grow differently from one living through repeated drought. Its immune system may face fewer infections. Damaged tissue can receive enough energy for repair instead of competing with the urgent demands of hunger or dehydration. Every body has a limited energy budget. During difficult conditions, immediate survival takes priority. Water must be conserved. Stored nutrients may be released to keep the organs working. immune activity and tissue repair still continue, though the resources available for them can become restricted. A safer environment changes that balance. Regular feeding supplies amino acids used to build proteins. Minerals support bone and shell maintenance. Stable temperatures allow digestion to proceed efficiently, rather than stopping through long cold periods. Veterinary treatment can remove parasites or control infections that would otherwise keep the immune system active for months. These improvements may allow more energy to remain available for cellular upkeep. The idea fits the broader life history pattern of tortoises. Their bodies evolved in lineages where adult survival already carried great value. When outside danger falls even further, maintenance systems may respond by preserving function for longer. The evidence does not show that zoo care makes an old tortoise young again. Cataracts do not disappear simply because food becomes dependable. Worn joints and damaged tissues can remain. The result concerns the rate at which mortality changes across a population. Several explanations also remain possible. Captive animals receive diets that may differ greatly from wild foods. Population density can change movement and social stress. Medical records are usually more complete, while the age and fate of wild individuals can be uncertain. Animals surviving long enough to enter a study may also represent an unusually durable group. These differences make direct comparisons difficult. Even so, the pattern challenges the idea of aging as one fixed schedule written entirely into the genes. The same species may produce different survival curves under different conditions. That possibility moves the explanation deeper into the body. Which tissues receive extra maintenance when food is steady? Do DNA repair systems work more effectively? Can inflammation remain under tighter control? The answers must be sought at a much smaller scale. Inside cells where strands break, proteins lose their shape, and tiny errors gather across the years. Inside every tortoise cell, small forms of damage appear during ordinary life. DNA can break when cells divide or when reactive molecules collide with it. A copying error may slip into the genetic code as a cell prepares to make two new cells. Proteins can lose their proper shape and stop doing their jobs correctly. Mitochondria, the structures that release usable energy from food, also produce reactive byproducts during normal activity. These unstable molecules can damage nearby proteins, fats, or DNA when they build up. The immune system creates its own risks. Inflammation helps contain infection and repair injured tissue. When that response remains active for too long, it can begin harming healthy cells around the damaged area. A body lasting for more than a century must manage these events millions of times. Cells contain repair systems that search DNA for broken or altered sections. Some enzymes remove damaged pieces and rebuild the missing sequence. Other systems reconnect DNA when both strands have snapped. Proteins that have folded incorrectly may be repaired by helper molecules or broken apart so their materials can be reused. Worn cellular parts can also be dismantled and replaced. When the damage becomes too severe, a cell may begin a controlled form of death called apoptosis. This process removes a dangerous cell before it can continue dividing or disrupt the tissue around it. Researchers have searched giant tortoise DNA for clues about how these maintenance systems may differ. One major project studied genetic material from Lonesome George, the last known pure Pinta Island tortoise. His genome assembly contained about 2.3 billion DNA letters. A genome is the complete set of genetic instructions stored inside an organism's cells. Scientists also sequenced an Aldabra giant tortoise and compared both animals with humans, mice, other reptiles, and several long-lived vertebrates. The comparison involved thousands of genes connected with development, immune defense, metabolism, stress response, cancer, and aging. Changes appeared in pathways linked with 6 widely studied features of aging. These included DNA damage, changes in metabolism, weakening protein maintenance, altered immune activity, shortening telomeres, and the loss of normal control over cell growth. Telomeres are protective caps found at the ends of chromosomes. They help prevent important DNA from being damaged each time a cell copies itself. One gene that attracted attention was called XRCC6. It helps repair double-strand breaks, which occur when both sides of the DNA molecule are cut. The giant tortoises carried a change in this repair protein at a position also altered in the naked mole rat. Naked mole rats are small African mammals known for unusually long lives and strong resistance to several age-related diseases. This shared change does not prove that both animals use the gene in exactly the same way. It provides a useful place for researchers to investigate. Another gene, DCLRE1B, contained a tortoise variant that may affect how its protein interacts with a structure connected to telomeres. Scientists also found signs of positive selection in genes called AHSG and FGF19. Positive selection means that certain inherited changes may have helped survival or reproduction enough to spread through a population over many generations. Both genes are involved in the way the body handles energy and chemical signals. Research in humans has also linked them with metabolic health and aging, although their exact role in tortoise longevity remains uncertain. A changed gene sequence is only a clue. An extra copy can provide more material for evolution to modify, but it may also become inactive. Laboratory tests are needed to learn whether a genetic difference changes the protein it produces. Researchers must then determine whether that change improves repair, reduces harmful inflammation, or meaningfully extends life. The tortoise genome, therefore, does not contain one clear instruction for living 150 years. It contains many possible adjustments spread across systems that protect, clean, and maintain the body. Jonathan's long life has depended on this work continuing quietly inside his tissues, day after day. Repair alone cannot solve every problem. A large body living through more than a century also gives damaged cells countless opportunities to ignore normal controls and begin multiplying. When a damaged cell begins dividing, each new copy carries the same mistake. Additional mutations can build on top of the first one. Some may weaken the signals that normally tell a cell when to stop growing. Others can help it avoid removal by the immune system. Over time, a small group of abnormal cells may form a tumor. This creates a special problem for giant tortoises. A large adult contains far more cells than a small reptile. Its body may also remain alive for well over a century. Each cell division creates a small chance that DNA will be copied incorrectly. More cells and more years should therefore provide more opportunities for cancer to develop. Simple mathematics would predict very high cancer rates in large, long-lived animals. Nature produces a more complicated pattern. Elephants contain many times more cells than humans. Some whales weigh more than 100,000 kilograms and can live for over 150 years. Giant tortoises may exceed 200 kilograms and survive through many human generations. These animals do not appear to develop cancer at the extreme rates expected from cell number alone. This puzzle is called Peto's paradox. It was named after British scientist Richard Peto, who drew attention to the weak connection between body size and cancer risk across different species. Within one species, a larger or older individual may face greater risk. Across distant species, size alone does not provide a reliable prediction. Evolution helps explain why. A lineage cannot become enormous and long-lived unless it also develops stronger ways to control abnormal cells. Individuals with weak cancer defenses would often die before completing their long reproductive lives. Over many generations, protective changes could spread through the population. Several layers of defense can work together. DNA repair proteins correct damage before a dangerous mutation becomes permanent. Tumor suppressor genes slow or stop cells that begin dividing at the wrong time. Immune cells patrol tissues and destroy cells carrying unusual chemical markers. A severely damaged cell may also begin apoptosis, the controlled self-destruction process introduced earlier. The giant tortoise genome study searched more than 400 genes already known to play roles in cancer. Researchers found extra copies or other unusual changes in several gene families linked with tumor suppression. These included genes known as SMAD4, NF2, PML, PTPN11, and P2RY8. Their normal jobs involve controlling cell growth, sending chemical signals, or helping damaged cells respond safely. Extra gene copies can sometimes strengthen a defense or allow one copy to develop a new function. A copied gene can also become inactive, so the number of copies never tells the whole story. One especially interesting result involved a gene called PRF1. Both giant tortoise genomes contain 12 copies of this gene. PRF1 carries instructions for making perforin. Perforin is a protein used by cytotoxic immune cells, which are immune cells able to kill infected or abnormal cells. They release perforin onto a target cell, creating tiny openings in its outer membrane. Other molecules then enter and help trigger the cell's destruction. Several PRF1 copies in Lonesome George were no longer functional. The remaining copies may still point toward an evolutionary history involving changes in immune surveillance. A duplication involving another gene, PRDM1, also suggested possible changes in how immune cells develop and recognize threats. These findings remain promising clues. Researchers still need laboratory experiments to learn whether the tortoise versions actually improve cancer resistance. A gene can look unusual in a DNA sequence, while producing only a small effect inside a living animal. Giant tortoises can also develop tumors, infections, and other serious diseases. Their genomes appear to contain several possible layers of protection, rather than complete immunity. That layered system makes biological sense for an animal built to last. A single checkpoint may fail. a later checkpoint can still stop the damaged cell. Repair proteins, growth controls, immune surveillance, and controlled cell death all reduce the chance that one mistake will spread through the body. Even these defenses cannot preserve every tissue forever. Jonathan's nearly 2-century-old body carries visible signs of age, revealing how slow aging can continue alongside gradual loss. Jonathan's condition today shows how several kinds of aging can unfold at different speeds inside one body. At an estimated age of at least 194 in 2026, he remains able to move across the grounds of Plantation House and respond to the people who care for him. Some of his organs have changed greatly during that long life. Cataracts have left him largely blind. A cataract forms when the normally clear lens inside the eye becomes cloudy. Light can no longer pass through cleanly, so shapes become blurred and vision gradually fades. Jonathan's sense of smell has also weakened. That loss matters to a tortoise because scent helps locate food lying close to the ground. His hearing remains much stronger. He can respond to familiar voices, movement, and the sounds associated with feeding. Caretakers often use sound to guide him toward a meal that he may be unable to see or smell clearly. These differences reveal an important detail about aging. The eye's nervous system, bones, muscles, and immune defenses do not all decline according to one shared clock. One organ can lose much of its function while other systems continue operating well enough to support daily life. The same pattern appears in many animals. A human may develop poor eyesight while retaining strong hearing. An old dog may move stiffly while its appetite and sense of smell remain sharp. Jonathan's unusual age stretches those uneven changes across a much longer period. By the late 2000s, his caretakers had become concerned about his physical condition. He was losing weight and finding it harder to feed effectively. His beak had become weakened by poor nutrition. A tortoise beak is made from keratin, the firm material also found in claws. It continues growing and wearing throughout life as the animal bites and tears plant material. When feeding becomes difficult, the body may receive too little protein, energy, and certain vitamins. Weakness can then spread through several systems at once. Caretakers began giving Jonathan supplemental food by hand. His meals included nutrient-rich vegetables and fruit chosen to improve his condition. Items such as carrots, apples, cucumber, bananas, and seasonal greens could be cut into pieces that were easier for him to take. The feeding sessions also allowed people to confirm how much he had eaten, rather than relying on food scattered across the lawn. His weight improved. The condition of his beak became better and his activity increased. An animal approaching 2 centuries of age had responded to ordinary nutritional support. That recovery provides a useful warning when studying very old bodies. A decline caused by poor diet, dehydration, parasites, or an untreated infection can look like the unavoidable final stage of aging. Correcting the immediate problem may restore function, even when the animal remains extremely old. Jonathan still carries permanent changes. His cataracts have not cleared, and his sense of smell remains limited. Other age-related wear may exist in joints, bones, and internal tissues that cannot be seen during a quiet walk across the grass. Slow senescence allows these changes to gather more gradually. it does not hold every part of the body in its youthful condition. A population can show a nearly flat mortality curve, even while individual animals develop sensory loss or need greater care. Demographic aging measures the rising chance of death across many individuals. It cannot describe every organ inside each member of that population. Jonathan's exact hatch date will probably never be known. The evidence supports an age of at least 194 in 2026, with the possibility that he is older. False reports of his death circulated online during that year. In April 2026, Guinness World Records confirmed that he was still alive on St. Helena. His continued survival depends partly on the biology that carried him through nearly 2 centuries. Regular observation and supportive feeding now help that biology continue. A long-lived animal can receive care from a community around it. A lineage reduced to one remaining member faces a very different limit, because even a body capable of another 50 years cannot reproduce alone. On Pinta Island, no caretakers were waiting beside the final tortoise of his line. The island lies in the northern Galápagos, where steep volcanic slopes rise above dry lowlands and scattered vegetation. By the early 20th century, the native giant tortoise was widely believed to have disappeared. Its scientific name was Chelonoidis abingdonii. For generations, visiting sailors had collected pinta tortoises for food. Later, introduced goats stripped away much of the vegetation that remained. With so few animals left, each death made recovery less likely. Then, in 1971, a Hungarian scientist named József Vagvölgyi was studying snails on the island. During his fieldwork, he encountered a living male tortoise. The discovery was unexpected because no confirmed member of the pinta population had been seen for years. Park staff later moved the animal to the Charles Darwin Research Station on Santa Cruz Island in 1972. He became known as Lonesome George. His age could never be measured exactly. Most estimates placed him above 100 years old. His shell, body size, and condition showed that he had already survived for many decades before anyone found him. George lived inside a protected enclosure where he received food, veterinary care, and regular observation. Those conditions could support his individual survival. They could not rebuild the population without another tortoise carrying the same pinta ancestry. Teams searched pinta for additional survivors. No confirmed female was found. George was later placed with females from closely related Galapagos populations. Researchers hoped that breeding might preserve at least part of his genetic line. Eggs were produced during some attempts, but none developed into surviving offspring that could continue the pure pinta lineage. The difficulty went beyond finding any available mate. A population needs enough males and females to reproduce across generations. Genetic diversity also matters. When only a few related animals remain, harmful gene variants can become more common, and the population becomes less able to respond to disease or environmental change. Suitable habitat must remain available for nests, hatchlings, feeding, and movement. Age groups need to overlap so that younger adults gradually replace older ones. George possessed only one part of that system. He was a living adult male. The rest of the population had already vanished. On June 24, 2012, Lonesome George was found dead in his enclosure. His death marked the end of the last known pure Pinta Island tortoise. He left no direct descendants. The extinction had unfolded slowly enough that one individual survived into the 21st century, even though the population had lost the ability to continue long before. George still contributed to science after his death. Preserved genetic material from his tissues helped researchers assemble the Pinta tortoise genome. That information later became part of the work comparing giant tortoise genes involved in DNA repair, metabolism, immune defense, and cancer control. His cells offered clues about how such animals can maintain their bodies for more than a century. They also preserved a record of a lineage that could no longer reproduce. Researchers have since found giant tortoises with partial Pinta ancestry, especially among animals connected to Wolf Volcano on Isabella Island. Their DNA suggests that Pinta tortoises bred with members of other populations after people moved animals between islands or carried them aboard ships. Those mixed descendants preserve fragments of the lost lineage. They do not recreate the original population that once lived and evolved on Pinta. George's story places longevity beside a much larger limit. An individual can survive for a century, while its species moves toward extinction around it. A durable shell protects one body. Slow aging can preserve that body through many seasons. Population survival depends on enough animals remaining together long enough to produce healthy generations. Pinta tortoises possess the biology for extraordinary life. Human removal worked on a much faster schedule, taking mature adults more quickly than a species needing 20 years or more to reproduce could replace them. By the late 18th century, ships crossing the Pacific had begun using the Galápagos as a place to gather food. Buccaneers had visited earlier, followed by sealers, whalers, naval crews, and merchant vessels. The islands offered little fresh water, but they held an unusually convenient source of meat. A giant tortoise could be carried aboard alive. Its body was adapted to uneven rainfall and long gaps between dependable food. Large adults stored water and energy, then lowered their activity when conditions became poor. Those same abilities allowed some captured tortoises to survive for weeks or months inside the dark hold of a wooden ship. Crews could keep them until fresh meat was needed. No barrels of salt were required, and the animal did not need daily feeding during much of the voyage. Ship logs sometimes recorded the number taken from individual islands. Tortoises were lifted, dragged, or rolled onto their backs for transport. Large adults were especially valuable because one animal could provide a great deal of meat. The loss of those adults caused the deepest damage to the population. Each one had required roughly 20 to 25 years to reach breeding age. A mature female might otherwise have produced eggs across many later decades. Historical reconstructions estimate that about 100,000 giant tortoises were removed from the Galápagos between 1774 and 1860. The true total may have been higher because many voyagers left incomplete records. Some animals died before reaching the ship. Others were collected by crews whose activities were never written down. Extraction also differed from island to island. Populations close to safe landing places could be reached repeatedly. A small group living on one isolated island had little ability to replace hundreds or thousands of adults taken within a few generations. The pressure continued after large-scale collection began to decline. Ships carried animals that escaped or were deliberately released. Settlers later brought livestock and domestic animals. Black rats entered nests and ate eggs. Pigs used their strong snouts to dig into nesting ground. Dogs could attack juveniles whose shells were still thin enough to damage. The adult shell offered no protection to an egg buried beneath a few centimeters of soil. Small hatchlings faced dangers that had been rare before human arrival. Vegetation changed as well. Goats ate grasses, leaves, seedlings, and low shrubs. Cattle trampled wet areas and competed for food. Donkeys opened trails and disturbed soil. These animals reproduced far more quickly than tortoises. A goat can begin breeding within its first few years and may produce young repeatedly. A giant tortoise waits 2 decades or longer before laying its first clutch. Pinta Island shows how quickly that imbalance could transform a landscape. In 1959, 3 goats were released there. By 1970, their population was estimated at about 40,000. The goats spread across slopes where Pinta tortoises had once fed. Young plants were eaten before they could grow. Ground cover thinned, and the remaining tortoises had fewer places to find dependable food. The goats were eventually removed through a large eradication effort. By then, the island had already been altered for decades. Its tortoise population had fallen so far that Lonesome George would become the final known pure member. The traits that supported a long natural life now worked against rapid recovery. Delayed maturity slowed the arrival of new breeders. Small clutch sizes limited replacement, especially when eggs and hatchlings continued disappearing. A population made of old survivors could appear stable for years, while quietly losing its future. Recovery therefore requires a schedule closer to the tortoise's own. Eggs must be protected through repeated nesting seasons. Young animals need years of growth before release. Restored adults may take decades to produce enough offspring for a population to support itself again. The work can begin within one human career, while the full result may belong to the people who continue it much later. On Pinzon Island, the oldest tortoises showed that adults could still survive. The missing age groups revealed the deeper problem. Black rats were eating eggs and newly hatched young before they could grow large enough to join the population. By the middle of the 20th century, researchers could find mature animals that had lived for many decades. Yet almost no juveniles followed behind them. Natural recruitment had nearly stopped. Recruitment is the addition of young survivors to a population. Eggs alone do not provide it. A hatchling must live long enough to become part of the growing population and eventually reach breeding age. In 1965, conservation workers began removing eggs from natural nests on Pinzon and carrying them to protected facilities on Santa Cruz Island. That operation became the first major captive rearing program for Galapagos giant tortoises. The aim was to protect only the most dangerous part of the life cycle. Eggs were taken away from rats and placed in conditions where temperature and moisture could be watched closely. After hatching, each young tortoise entered a long period of supervised growth. Workers recorded body weight and measured the shell. Identification marks allowed each animal to be followed through time. Today, tiny microchips can be placed beneath the skin, giving every tortoise a permanent identification number that can be read with a scanner. The young animals receive suitable food, shelter from temperature extremes, and protection from predators. Their shells gradually become thicker. Leg muscles strengthen as body mass increases. Water and energy reserves grow large enough to support longer periods without feeding. Most remain in protected care until around 5 years of age. The exact release age can vary because islands contain different predators and habitats. By roughly 5, many juveniles have crossed the stage when a rat or small bird can kill them easily. In 1970, the first 20 captive reared Pinzon tortoises were returned to their island. They had spent their earliest years away from the danger that had removed nearly every wild hatchling. Later groups followed. This approach became known as head starting because it gives young animals a protected beginning before they resume life in the wild. The process begins even earlier for populations maintained through captive breeding. Adults are paired using information about species, ancestry, and genetic diversity. Genetic testing helps managers avoid mating animals that are too closely related. Eggs are placed in incubators, where temperature can influence the number of males and females produced. Warmer conditions usually create a greater proportion of females. A recovery program may use that effect when a population needs more egg-laying adults in future decades. Incubation still requires care because excessive heat can damage embryos. Managers work within temperature ranges suited to the particular tortoise population. Once the hatchlings emerge, growth records continue for years. Before release, animals may pass through quarantine. This period allows staff to watch for signs of disease and reduces the chance of carrying parasites or infections into a wild population. Transport to an island can involve boats, aircraft, crates, and teams moving carefully across rough volcanic ground. The release site must offer suitable food and shelter. It also needs protection from the original cause of decline. Head starting cannot rebuild a population if rats continue consuming every new generation after the program ends. On Pinzon, rat eradication in 2012 allowed wild hatchlings to survive again. The protected rearing program carried the population through the years before that larger habitat problem could be solved. 3 main tortoise breeding centers now operate on Santa Cruz, Isabela, and San Cristobal. Across several decades, more than 10,000 captive reared giant tortoises have been returned to native habitats in the Galápagos. Some released animals are now old enough to reproduce on their own. Their nests can begin replacing the work once performed inside incubators. The scale of recovery remains incomplete. Living Galápagos giant tortoises are estimated to represent only about 10% of their historical abundance. They occupy roughly 35% of the habitat that could potentially support them. Continued progress depends on invasive species control, connected migration routes, and years of field monitoring after release. 10,000 returning tortoises represent thousands of carefully protected beginnings. Each one also becomes part of the physical island, pressing paths into vegetation and carrying seeds through a landscape that developed around giant herbivores. Once released, a giant tortoise begins changing the ground around it. Its beak clips grass and low leaves. A heavy shell pushes through shrubs, while broad feet press narrow tracks into the soil. Low branches may bend or snap as the animal passes beneath them. seeds swallowed with fruit can travel through the digestive system and return to the ground inside dung, sometimes far from the plant where the journey began. That dung also carries nutrients. Nitrogen, phosphorus, and other materials taken from plants are moved into new parts of the landscape. Rain can wash some of those nutrients into the soil, where roots and tiny organisms use them again. Animals that change habitats through their ordinary behavior are called ecosystem engineers. A giant tortoise performs that work by feeding and moving. Its pathways can open dense vegetation. Shorter plants may grow where repeated grazing allows sunlight to reach the ground. Seeds can arrive in places they might never have reached through wind alone. One adult may continue these actions for more than a century. A trail can be crossed year after year. The same feeding ground may be cropped across many rainy seasons. Longevity therefore gives a tortoise more time to reproduce and more time to shape the island that supports it. This role became especially clear on Floriana, one of the southern islands of the Galápagos. Native giant tortoises disappeared from Floriana during the 19th century. Sailors removed adults, while introduced animals damaged nests and vegetation. For more than 180 years, the island lacked the large herbivore that had once moved seeds and opened paths across its slopes. A return began on February 20th, 2026. That day, conservation teams released 158 young giant tortoises carrying Floriana ancestry. The animals were between 12 and 14 years old. They had already passed through the most vulnerable years of early life, yet they were still young by giant tortoise standards. Many may remain on the island long after the people who carried them there are gone. Their ancestry had been recovered through genetics. Decades earlier, researchers found unusual giant tortoises living around Wolf Volcano on northern Isabella Island. DNA testing revealed that some carried genes from the lost Floriana lineage. Their ancestors had probably been moved by sailors, then released or abandoned among tortoises from other islands. Selected adults were brought into a breeding program. Pairs were chosen to increase the amount of Floriana ancestry passed into later generations. The young animals released in 2026 came from that careful process. They are descendants of a lineage once considered completely lost from its native home. The release was timed for the rainy season. Fresh vegetation was more abundant, and temporary pools offered water. Those conditions gave the young tortoises a gentler beginning as they explored unfamiliar ground. Each animal carried a lightweight GPS transmitter. The devices can record movement and activity every hour. Researchers will be able to see which valleys the tortoises enter, where they spend the hottest parts of the day, and how quickly they spread from the release area. Their routes will reveal more than location. Scientists can compare movement with plant growth, rainfall, elevation, and water availability. Field teams can watch for changes in vegetation, where tortoises begin grazing regularly. Dung can be examined for seeds. New pathways may show where repeated travel is opening the landscape. The wider Floriana restoration effort also includes removing invasive mammals and repairing damaged habitat. Other native animals may eventually return once food webs and nesting areas become more stable. The work will take years because ecological recovery moves through breeding seasons and generations. A modern threat remains difficult to plan around: Rainfall patterns are changing, and hotter conditions can affect plant growth, water supplies, and nest temperatures. One tortoise released at age 12 could still be alive late in the 22nd century. During that lifetime, its island may experience environmental changes far faster than those that shaped its ancestors. The success of a durable body still depends on a landscape that continues providing food, water, nesting ground, and connected roots. Floriana now holds living evidence of that relationship. A long lifespan grows from many protective systems working inside the animal, while the island around it supplies the conditions those systems need. The young tortoises on Floriana carry many layers of history inside their bodies. Their shells come from an ancient structure that began forming more than 250 million years ago. That armor lowered the chance that a healthy adult would be killed by an ordinary predator. Greater adult survival made later breeding seasons more valuable. Over long periods, natural selection could favor animals that grew slowly, delayed reproduction, and maintained the same body for many decades. The shell created the opportunity for longevity. It did not control every process required to achieve it. DNA still had to be repaired. Damaged proteins still needed to be removed. Infections still had to be contained before they spread through the body. Energy also shaped the schedule. As ectotherms, tortoises rely strongly on heat from sunlight and the surrounding air. They spend far less food energy maintaining one high internal temperature than a mammal of similar size. Their metabolism changes with weather, activity, recent feeding, and body mass. That flexibility allows long periods of rest during drought or extreme heat. It also ties survival closely to shade, water, seasonal plants, and suitable ground temperatures. Many other ectotherms live only a few years. Environmental heat therefore supplies one part of the explanation rather than a complete rule. Large size adds further protection. A giant adult can hold more water and stored energy than a hatchling. Its temperature changes more gradually, and few natural predators can threaten it once the shell has fully developed. Reaching that size may require more than 20 years. Long adulthood then gives a female repeated chances to nest, even when drought, flooding, or predators destroy the young from one season. Migration connects those reproductive years to the landscape. Highland feeding grounds provide energy. Lowland soil provides warm nesting sites. Open routes allow tortoises to move between them as rainfall and vegetation change. A blocked corridor can weaken that entire system, even when both ends of the journey remain protected. Population records reveal the result of these combined advantages. In the 2022 study of 52 turtle and tortoise species, roughly 3/4 showed slow or negligible senescence. Their risk of death increased very little across much of adulthood. Those records describe a broad survival pattern. They do not identify which molecule keeps one animal alive. Genome research offers possible clues at that smaller scale. Giant tortoises carry unusual changes in genes, connected with DNA repair, metabolic control, immune defense, telomere maintenance, and tumor suppression. Some may improve the detection of damaged cells. Others may help regulate inflammation or control abnormal growth. These pathways remain research candidates. Laboratory experiments are still needed to learn how much each genetic difference contributes to a real tortoise lifespan. they are also far from becoming treatments for human aging. The line leading toward humans separated from the tortoise lineage more than 300 million years ago. Our bodies maintain a high internal temperature, grow on a different schedule, and reproduce under very different biological pressures. Tortoise research can still teach useful principles. Scientists can study how cells respond to DNA breaks, how immune cells recognize abnormal tissue, and how long-term inflammation is controlled. They can examine protein maintenance, energy use, and the effect of safe environments on the rate of senescence. Those lessons may guide future research without turning a tortoise gene into a simple human cure. Every part of the longevity system has limits. A shell cannot protect an egg from a rat. Strong cellular repair cannot replace a missing mate. A century-old adult cannot rebuild a population when migration routes disappear and hatchlings fail to survive. The Floriana release required genetics, captive breeding, invasive species control, and habitat restoration, because biology works inside an environment rather than apart from it. Jonathan's life on St. Helena now looks different from the way it appeared at the beginning. His age does not come from one hidden substance. It reflects protection, careful energy use, slow growth, cellular maintenance, and years of supportive care. His eyes have developed cataracts, his sense of smell has weakened, and regular feeding helps him remain in good condition. Time continues changing his body. Across nearly 2 centuries, however, damage has usually been repaired, contained, tolerated, or survived before it became fatal. On the lawn at Plantation House, Jonathan lowers his head toward the grass. Sunlight warms the broad surface of his shell. His heart continues moving blood through tissues first formed in a world without airplanes, antibiotics, or electric homes. The tortoise does not escape the passage of time. For an unusually long while, its body continues finding enough ways to carry on.